Chromia.
A gentleman would at least offer to buy me a drink first.
Norman, pint of bitter for me and a brandy for the lady, please.
How did you know that?
Lucky guess. Name's Xavier. Charles Xavier. How do you do?
Amy.
Heterochromia was in reference to your eyes, which I have to say are stunning. One green, one blue. It's a mutation. It's a very groovy mutation. I've got news for you, Amy. You are a mutant.
First you proposition a girl, and then you call her deformed. How is that seduction technique working for you?
I'll tell you in the morning. No, seriously, though, you mustn't knock it. Mutation took us from single-celled organisms to being the dominant form of reproductive life on this planet. Infinite forms of variation with each generation, all through mutation.
Then let's reclaim that word. Mutant and proud.
Chin chin.
Hello everyone and welcome to chapter 6.2b. This is the last part of chapter 6 and it's about mutations. Now, I know you saw the video clip just now and it's incredibly fascinating, but guys, please do not act like Charles Xavier. It's incredibly arrogant and annoying. So, okay, don't do that. However, Charles Xavier is right about mutations. You know, mutations are not always harmful. In fact, they're incredibly important and helpful in the process of evolution. And that's because mutation results in variation. Mutation is what gives rise to new alleles. But you know, to unpack that a little bit further and understand mutations more, we need to define it, okay, and need to understand the processes. So mutations in general is a random change in nucleotide sequence of a gene. And this could affect the phenotype. Phenotype means trait or appearance of an organism. Now, there are two major types of mutations, two levels, I would say. One is a chromosomal level and one is a gene level. Chromosomal mutation will affect the structure or the number of chromosomes, and actually, because if you think about it, you know, chromosomes code for many genes. So a change in structure would affect many genes. So it's a very large scale mutation here. It's very problematic. However, that's not the syllabus. We are mainly focusing on gene mutations. So mutations that only affect a single gene here. So let's define it in regards to gene mutations. Gene mutations, are random changes in a sequence of bases in a gene. And this causes altered codons in mRNA sequence, okay, because, well, if the DNA codes for the wrong thing, the mRNA is going to code for the wrong thing as well. And therefore, this alters the amino acid chain sequence. This can result in new alleles, and this is what Charles Xavier is talking about. This is why mutations are so important, because it results in new alleles, which gives variation, which later on we'll see in A2 how it drives evolution. Alleles is defined as different forms of one gene, by the way, and this is an important definition to remember. You will see this definition popping up in your syllabus every now and then and ask more questions. Anyways, it's important to note that gene mutations are not only important for forming new alleles, mutations are actually really common in human beings and in every living organism. If you think about it, we are exposed to many mutagens all around us all the time. But we do not always suffer harmful effects. And that's because there are many different types of mutations. And it has to happen in a specific location, in a specific sequence, in a specific way in order to cause harmful effects. So let's understand it more by looking at the types of different gene mutations and its consequences. So we are looking at substitution, insertion, and deletion. So substitution is obviously just replacing the base. So G change to C is substitution. Insertion is addition of a base. Deletion is removal of a base sequence. And in our syllabus, we only look at the simplest form of mutation, okay, which is just a change of one nucleotide. And a change of one nucleotide is often referred to as base mutation or point mutation. So yeah, these are the different types of point mutation we're going to look at, starting with base substitution. So substitution at a single base only. Again, substitution means that one nucleotide is replaced by another, and this can result in one of three types of mutations. And these names here are mostly describing the consequence of that base substitution. So it could be a silent mutation, which is great because by changing the base, by substituting the base, the triplet code and codon still so happens to code for the same amino acid. And as we learned in the previous part, multiple triplet codes can code for the same amino acid because the DNA triplet code is degenerate. And therefore, if this happens, this kind of substitution happens and results in a silent mutation, this would not affect the amino acid sequence at all. However, sometimes, right, just so happen that base substitution could potentially result in a nonsense mutation. And the nonsense mutation is basically when a stop codon is introduced. Now, the third consequence of base substitution could be, depending on the location, right, missense mutation. And this really refers to the fact that the triplet code codes for a different amino acid. So what is the consequence of these different mutations on the protein? So depending on the location of the base substitution, okay, and the sequence, if it's silent, it's gonna cause the triplet code to change. However, the amino acid sequence is unaffected, so the protein is normal and functional. Now, what if it's nonsense mutation, right? So you can see here TTC became ATC, and you can see the codon change as well, UAG, which is a stop codon. What would happen then to the amino acid chain, right? So this would result in premature chain termination. So every triplet code, every codon after that is not read and subsequent amino acids are not formed. And therefore, you end up with a shorter, truncated version of the protein, which is incomplete and non-functional. This is very problematic. Now, again, depending on the location of base substitution, this could also result in a missense mutation, as we saw just now. TTC become TCC, or maybe TTC would become TGC. And in both cases, lysine is changed to a different amino acid. However, we see here that lysine and arginine are both in a purple box, which tell you that they're both basic. Threonine, on the other hand, is polar. Now, how would this affect the polypeptide chain? Now, missense mutation, as we know, causes one amino acid of the polypeptide sequence to be affected. So one amino acid is changed only. It's not a stop codon where all the other subsequent amino acids are not formed. Only one amino acid is affected here. Lysine become arginine, lysine become threonine. Everything else in the protein chain is fine. However, the property of the amino acid does matter, because if the amino acid has a side chain with a different property, the tertiary structure will be more affected, because different property means it can form different bonds, and that means that the coiling and the folding of the polypeptide structure would be different, and tertiary structure, 3D structure, will be more affected. This would result in an overall faulty protein, which may be functional. So there is a maybe there. It would be more likely be functional if the amino acid resulting is the same property. Okay, there is a possibility, but if it's a different property, then it's less likely to be functional, but there is still some probability there. Now, if this is an enzyme we're talking about which is affected, and you change a very essential amino acid that is at the active site, then the faulty protein will definitely be not functional. So it really depends which amino acid is affected as well. If that amino acid happens to be important to the function of the protein, then, well, too bad, the protein is not going to work. However, if the amino acid change is not that essential to its function, then maybe the protein will still be able to work. So yeah, that's the different consequences base substitution can result in. It's either going to be silent, or it's going to be a nonsense mutation, or a missense mutation. And depending on the location of that mutation and what depends on the protein as well, it would have different effect on the polypeptide chain. In the exams, these kind of questions are usually quite context specific, so make sure you read the question carefully and identify the mutation yourself. Okay, so that's base substitution. Let's look at the effect of base insertion or base deletion, and both of these actually result in the same effect. Because what it results in is frame shift mutation. This is a big word. Remember this word, frame shift. What do you mean by frame shift? It's an important concept to understand, but you're not required to describe it that much. So frame shift is when the reading frame shifts. Okay, what is it, reading frame? Reading frame is, you know, how three bases are read together. So CAC, 3 bases are read together, which codes for histidine. TGT, read together. TAC, read together. GGT, read together. This would be called the reading frame. However, if you delete G here, this shifts the reading frame. So we call that frame shift. So instead of TGT, the 3 bases that are read together is TTT, and then ACG, and then GT something else, which is not written here. But as you can see, it seems like the reading frame is shifted like 1 base forward. This results in all subsequent codons being affected. Whether it's an insertion or deletion, this results in a frame shift. Everything else after the mutation, every codon are affected, and therefore every subsequent amino acid after the point of mutation would be affected. Now, because of this drastic change in reading frame, this could also result in a nonsense mutation. And this means that a stop codon could occur, right? Deletion or insertion of one nucleotide could cause the reading frame to shift in such a way that maybe somewhere along the line, a stop codon is reached prematurely. Okay, either way, for insertion-based deletions, this results in non-functional polypeptides and affects a lot. Okay, affects everything after that mutation. So yeah, again, insertion and deletion, right? This results in frame shift, which could also result in a nonsense mutation. Okay, I guess the question I get sometimes is, what if 3 nucleotides are deleted at once, or 3 nucleotides are inserted at once? Now, I guess if you insert 3 nucleotides at once, depending on where you insert it, this would probably make the damage quite localized. So it can result in the deletion of 1 codon, or just deletion of 1 amino acid, or addition of just 1 amino acid. Or maybe 2, depending on where it's inserting into or deleted from. Okay, the damage will not spread to all the subsequent amino acids. So it's probably best if 3 nucleotides are deleted or inserted at once instead of just 1, even though there's more nucleotides, but all the subsequent amino acids may not be so affected. And protein may be still functional, yeah. Okay, anyways, one more thing. We need to learn about sickle cell anemia. This is an example of a base substitution mutation at the gene coding for beta globin in hemoglobin. Now, as we have learned in previous chapters, hemoglobin is made out of 4 polypeptide chain, 2 alpha globins, and 2 beta globins, right? This base substitution affects the 6 amino acid of beta globin. Now, what happened here? For every mutation question, you should be following this kind of structure, right, in describing. You need to go through all the different levels here. Level 1 is DNA. What happens at a DNA level? If the DNA changes, this is considered a mutation, right? So here, CTT is changed into CAT. So A replace T. If you're using the textbook, especially the green color one from Cambridge, the textbook has a typo and is wrong. And yes, you do need to remember that CTT is converted to CAT. Okay, you need to remember that this is the mutation. Now, what happens here is an mRNA codon that's different is formed. GAA changed to GUA, and as a result, different tRNA brings a different amino acid to the ribosome, leading to a change of amino acid in the polypeptide chain, altering its primary structure. Now, specifically, glutamic acid, which is what it's supposed to be, right, has changed to a valine. Glutamic acid is polar, whereas valine is non-polar. And therefore, since they have different properties altogether, this changes the secondary, tertiary, and quaternary structure of hemoglobin, resulting in many problematic effects. What is it? Mutated beta globin causes, number 1, hemoglobin molecule to be less soluble. HB stands for hemoglobin here, right? Less soluble, and therefore they clump together and stick together to form fibers within the red blood cell. Okay, this hemoglobin is clumping within the red blood cell. You can see the diagram at the right hand corner here, and therefore they are less able to bind oxygen as well. And because of those fiber formation, this causes cells to become more crescent or sickle shaped. Okay, and this sickle-shaped red blood cells are honestly more prone to rupture and more prone to block blood vessels. And as you can see, if it's round, it can go through with no problem, but when it's sickle-shaped, it can block vessels in this formation right here. And this would cut off blood supply and would be very detrimental. Now, however, in order to experience symptoms like this and really experience symptoms of sickle cell anemia, that person must inherit 2 mutated beta globin alleles, one from the mom and one from the dad. So we can see affected daughter here. This daughter affected would have 2 copies of the sickle cell allele. So the mutated beta globin alleles is represented by HBS. This is like the official way of representing it. This is a diploid organism. So 2 alleles for each gene, 2 sickle cell anemia, 2 mutated beta globin alleles are needed in order to get this disease. Again, one from father, one from mother, which means both parents have to be carriers. Okay, or one affected and one's a carrier. So what's the carrier's genetic composition? Should be HBA, which is normal, HBS. The mom is also HBA, HBS. So 1 normal allele, 1 mutant allele. The daughter inherits this allele and this allele to get 2 alleles and form the disease. Now, what is with carriers? So how do carriers actually not experience the disease? So the thing is, they do have 1 working copy, 1 normal one, and 1 one non-functional one, and therefore because of this they have no disease, but they do carry the sickle cell trait, which means when you look at their red blood cells under the microscope, you will still see some cells have the sickle cell trait. They are sickle shaped. However, they do not experience the disease because, well, they still have 1 working copy, 1 allele that is perfectly fine. So they do not experience the symptoms at all. They have enough functional beta globin alleles in order to make functional hemoglobin, which allows them to function normally. So yeah, that is sickle cell anemia for you. And with that, we have pretty much come to the end of the chapter. Now, before we go to end, I just have a few closing remarks and reminders for gene mutations. Again, when you talk about gene mutations, make sure you read the question carefully. More often than not, they usually give you the context and tell you the disease, tell you what the mutation is. You need to make sure you talk about every single level. If they ask you, so how does this mutation affect the polypeptide? Make sure you talk about what happens at the DNA level. Try to be as specific as possible. What happens at the mRNA level? Say that if it's changed tRNA, what happens to tRNA? Does it bring a different amino acid? And what happens to a polypeptide? Are its levels or functions affected, right? Is it functional? Is it non-functional? Is it complete? Or is it incomplete? So yeah, every single level needs to talk about here. Go through the flow chart and you should be just fine. But yeah, that's it for mutations. I'll see you next chapter. Bye-bye.